Think of the happiest thing you can.
The DNA. So the DNA is a long polymer of deoxyribonucleotides. Okay, they are telling that many nucleotides, many deoxyribonucleotides join together to form deoxyribonucleic acid. That is why deoxyribonucleic acid is a polymer of deoxyribonucleotides. So the length of DNA is usually defined as number of nucleotides. Okay, so we know that it is a collection of nucleotides, and we are counting the number of nucleotides which is present in an entire deoxyribonucleic acid, and it is defined on that basis. That is, the length is defined on that basis. Or a pair of nucleotide. Okay, so two nucleotides joined together is known as base pairs. Okay, like DNA will be there like this. Okay, two strands will be combined together, like here will be one nucleotide and here will be another nucleotides. So these two opposite nucleotides will join together to form one base pair. So they are telling that either the length of DNA is defined in terms of separate nucleotides, or it's defined in terms of the base pairs. Okay, so for example, if you see bacteriophage, which is known as 5 into 174, it has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs. So here you can see that it is defined in terms of individual nucleotides, and here it's defined in terms of base pairs. And Escherichia coli has 4.6 into 106 base pairs, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. It's not 106, it's 10 to the power 6, and this is 10 to the power 9. Okay, so this is very important. The haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. Okay, so the thing is that this length of DNA is characteristic of an organism. That is, according to the organism, the number of nucleotide which is present in the DNA will vary. Okay, so now we are going to discuss about the structure of a polynucleotide chain. Okay, so a polynucleotide chain, it can be either DNA or RNA. So first of all, if you see a nucleotide, it has three components: a nitrogen base, a pentose sugar, and a phosphate group. So first, if you consider the nitrogen base, okay, there are two types of nitrogen bases: purines, which is adenine and guanine, and then pyrimidines, which is cytosine, uracil, and thymine. Okay, so here you can see the structures of the nitrogenous bases. This is adenine, guanine, cytosine, thymine, and uracil. So you can ask, what is purines and pyrimidines? Okay, purines, it contains 2 carbon nitrogen rings and 4 nitrogen atoms, and pyrimidines contain 1 carbon nitrogen ring and 2 nitrogen atoms. So here you can see in the purines, which is adenine and guanine, there are 2 rings, right? Here is 1 ring, and then there is another ring in here. Okay, so there are 2 carbon nitrogen containing ring, and there are 4 nitrogen atoms. Here 4, and here also there are 4 nitrogen atoms. And pyrimidines, it contains 1 carbon nitrogen ring, and it contains 2 nitrogen atoms present in the ring. One thing which you have to remember is that cytosine is common for both DNA and RNA. Okay, when you consider pyrimidines, cytosine will be present in both DNA and RNA. Thymine will be present in DNA, and in the place of thymine, uracil will be present in RNA. So thymine, since it is T, you remember it as it will be there in DNA, and uracil will be present in RNA. So purines, adenine, and guanine are common for both RNA and DNA. So coming to our next component, which is pentose sugar. ribose will be present in RNA. That is, ribose is a pentose sugar which is present in RNA, and deoxyribose is a pentose sugar which is present in DNA. So here are the structures of ribose and deoxyribose. So this is called deoxyribose because there is one oxygen atom, okay, which is actually missing here. So at the second position you can see that there is one oxygen atom missing when compared to ribose. So that is why it is called deoxyribose. Otherwise the structures of ribose and deoxyribose are quite similar to each other, okay. And usually this pentose sugar is numbered, okay. The carbon atoms which are present in the pentose sugar is numbered as 1, 2, 3, 4 and 5, and since there are 5 carbon atoms, this is known as pentose. Since there are 5 carbon atoms, it is called pentose sugar. Sugar because it is carbon containing carbohydrate, and hence it is also known as sugar. So you will be learning about it in detail in your biomolecules chapter. So last is phosphate group. Phosphate group do not have much variations when compared to DNA and RNA. So components of nucleoside, first is nitrogen base. We have purines and pyrimidines. In purines we have adenine and guanine, which will be present in both DNA and RNA. And coming to pyrimidines, we have cytosine, uracil and thymine. Cytosine will be common for both DNA and RNA. Thymine will be present in DNA and uracil will be present in RNA. Coming to pentose sugar, ribose will be present in RNA and deoxyribose will be present in DNA. And last is phosphate group, it will be present in both RNA and DNA.